During Clathrin-mediated endocytosis, a polyhedral lattice of clathrin is linked to membrane proteins or lipids via clathrin-binding adapter proteins, such as SLA1. SLA1 binds clathrin through its variant clathrin box, or VCB. In this video, three methods are used for analysis of clathrin-SLA1 interactions. First, yeast cells expressing GFP-tagged native and VCB mutant SLA1 are imaged using spinning disc confocal microscopy. Time-lapse movies are then analysed to determine the effect of mutation on the kinetics of endocytosis. Second, yeast cytosolic extracts are prepared from wild-type cells as a source of clathrin, and GST-VCB fusion protein pull-down assays are performed to examine Clathrin-VCB interaction in vitro. Third, total extracts are prepared from yeast cells expressing native and VCB mutant SLA1, and co-immunoprecipitations are performed to examine the effect of mutation on the affinity of SLA1 clathrin binding in vivo. Ultimately, the data obtained show that mutation of the VCB delays endocytosis, and that binding of SLA1 to clathrin is lost in these mutants.
Though this method can provide insight into clathrin adapter dynamics in endocytosis, it can also be applied to other protein-protein interactions within vesicles in the secretory pathway.
Generally, individuals new to this method will struggle because photobleaching can dim the fluorescent signal from the lower expression of tagged proteins from an endogenous locus.
Because this protocol combines image analysis with